Hi everyone, my name is Miranda and I am a graduating senior here at Meredith this year. I completed my project this past summer at the Duke University Marine Lab in Beaufort, North Carolina. This was a part of the RAU internship that I was able to get into. And my project is the Megalopae molting of the Euca species Fiddler crabs. Do habitat and adult odor cues affect the timing? My advisors for this were Doctors Kathy Reinsell and Jim Welch. And let's get started. So the objectives of my project were to see, do habitat odors affect where megalopae grow and how quickly they molt into juvenile crabs? And do adult odors affect where megalopae grow and how quickly they molt into juvenile crabs? So a little bit of background on this, if you're not familiar, there are 3 species of Euca fiddler crabs along the Atlantic coast. There is the Euca pugilator, which is also called the sand fiddler. That is that top picture. They are known for their carapace having a purple top, and the carapace is that hard shell in the middle. The next species in the middle picture is the Euca pugnax, which is also called the mud fiddler. They are known for having yellow claws, and sometimes they have a blue stripe along their carapace, and they are bigger than the pugilators. And lastly, the Euca minax, which is also called the red-jointed fiddler in that bottom right picture. They're known for having red joints and for their large size, so this is the largest of the 3 species. All of the species are identical at the larval and juvenile crab stages, so you can't really tell which species is which. The megalopae is the 3rd stage of the life cycle right before they molt into the juvenile crab, and the megalopae live in open waters and settle in certain habitats in order to molt into juveniles and then into adults. So this slide is basically showing the life cycle, and in that picture, that is the actual megalopae, one of them that I worked on during my experiments. So an adult female will hold on to the eggs until they're ready to hatch. The zoea are pulled out into the ocean with the night ebb tides, which is the low tide, and they are carried into the ocean where they eventually molt into megalopae. Those megalopae use nighttime flood tides, which are high tides, to move towards the shore in order to settle. And once they settle in a habitat, they molt into juvenile crabs and eventually into adult crabs. So it's not really known whether or not megalopae can choose their settlement site specific for their species or if it's just random. There have been prior studies that have shown that the odor cues of other adult Euca species crabs can affect the molting process. It's also been shown that if the odor is of the same species, then the molting time for those megalopae is a lot shorter. And for those of you who don't know, those are both Euca pugilators in that picture. The way we tell the difference between a male and a female is that the males have the one larger claw and the one smaller claw, and then the females have 2 smaller claws. The males use their larger claw in order to seduce the females to go into their burrows, so they do a waving motion with it. And they can also use it for fighting other males. So my experimental design, over my time at the Marine Lab, I conducted 4 experiments that each lasted 10 days. And within those experiments, there were at least 120 megalopae tested in each experiment, which is all of the different tubes that you see in that bottom picture. So my daily process, every day I would go and collect sand from a small beach in downtown Beaufort, and I would just take the first few millimeters off the top of the sand. And we chose this beach because there was a presence that we had seen of Euca species there. So we knew there were adult odors already in the sand. Excuse me. So I would collect the sand and bring it back, and then I would collect water off the dock and check the salinity, which was normally 35 parts per thousand, unless it had rained or stormed badly the night before. And that water was filtered into a flask using a cloth filter, which that ended up being the controlled seawater. The other odor waters were sand, crab, and crab and sand. The sand consisted of 25 grams of sand. The crab water consisted of 25 grams of pugilators that we had already collected before. And the crab and sand consisted of 25 grams of pugilators and 25 grams of sand. So once all the water and the sand and crabs were placed into their flasks, they would be incubated with bubblers for an hour. So basically, a long tube would be blowing bubbles and air into the water, which stirs everything up and makes all the odors get into the water. Once all that was done, I would decant the water through a sieve, which is basically just pouring the water from the flask through a filter, and it just keeps the sand and crabs from flowing into the water. Once that was done, I would place the water in the vials and then place the megalopae that had been collected into the different vials. They would be changed every day, and I would have to make new water treatments every day. They would also be fed Artemia because they still eat, which are brine shrimp. So each day I would check for molting 4 times, one before breakfast, at lunchtime, right after dinner, and right before bed. And so what I would be checking for to see if they had molted is to see if their body had become a lot wider and if they were not doing as much swimming and they were doing more walking on the bottom of the vial, since megalopae are known to swim and their bodies aren't as wide. After each experiment, I would do a DNA analysis to determine the identities of each animal, since they all look the same. So what I would do is a DNA extraction, PCR, and gel electrophoresis. And then once all of that was done and each species was determined, I would use statistics to determine the relationships between the different species and the different water treatments. So this is a picture of me in the process of doing PCR. This is what a gel electrophoresis looks like, well, not the machine, but the gel after it's run. So this points out the bands of each species. At the end, all the way to the right, there are 3 bands on the top and bottom. Those are the primers that have been used to determine each different species, so they line up across. So the bottom one, the lowest one is Eucopugilator, the middle one is Eucopugnax, and the highest one is Eucominax. So this would be how I would determine which species each one was. So Eucopugilators. The proportion of pugilators that molted in each treatment was higher than the amount of remaining megalopae. All of the crabs and the crab and sands molted, and there were actually no megalopae left at the end of the experiments. So when compared to each other statistically using Z-tests in Microsoft Excel, the control and sand were not significantly different from each other. That's why they both have the A's. The control and the crab and sand were significantly different from each other, so that's why they also have different letters. And the sand versus the crab versus the crab and sand versus sand, and then the crab versus the crab and sand were not significantly different from each other, so that's why they all have C's at the top of their columns. And so basically, again, this shows that all of the Eucopugilators molted in all of the water treatments, and but the most that molted were in the ones that had adult odors. So this graph shows the average time that it took to molt in hours for each species in each treatment. The numbers in each of the bars represents how many molted in that specific water treatment. And it shows that the crabs and the crab and sand water treatments had the most megalopae that molted, and they took the shortest amount of time to molt. And overall, the Eucopugilators that molted tended to change within the first few days. So Eucopugnax, the control versus sand are not significantly different from each other, so that's why they had the A's above them. The crab versus the crab and sand were not significantly different from each other, which is represented by the B's. And then all of the rest compared against each other are significantly different, so that's why they don't have repeating letters above them. And the Eucopugnax also grew more in the presence of the adult odors, even though it was pugilator water. And the habitat didn't really seem to have much of an effect on the megalopae. So the average time to molt for Eucopugnax, it took them longer than pugilators to molt, and they would change more towards the middle of the experiment than the beginning. And this also shows that the crab and sand had the most molts, and the megalopae molted faster in the ones that had adult odors in them. Eucominax, the minax didn't really molt to any of the odors, whether it be habitat or adult, but it did end up, the ones that had molting in them, the most happened in ones that had adult odors. Majority of the Eucominax stayed megalopae through the entire project. They are very picky compared to the other species. So that's why they didn't really have as many megalopae molt. And there was no significant difference between any of the water treatments. The average time it took for Eucominax, they took the longest of all the species to molt. The few that did molt changed towards the end of the experiment and only had around one of the megalopae to molt into crabs, juvenile crabs, except for the crab treatment, which had 5. All of the graphs have supported the prior studies that they are correct about the fact that adult odors affect the timing of molting and how many megalopae will molt. And so far, like I've shown in all the graphs, nothing really proves too much about the habitat having any effect on the process. It might have a slight effect since some of the megalopae did molt in the habitat ones, but it's not as prominent as the adult odors. So the conclusions, basically, like I said, the adult odors made it faster for the megalopae to molt and increased the number that molted. All the species reacted to the eucopugilators. And the control water had the longest amount of time to molt and the least amount of megalopae that molted. So there were some problems that occurred, because there's always problems with research. One of the problems was that there were only specific times to set out the plankton net in order to catch megalopae, because they do come in with the nighttime tides, so I had to wait for those. There were ctenophores, which are somewhat like jellyfish. They eat megalopae and catch them in their slime. We also had a theory that the megalopae might have been staying more towards the lower part of the water column, since the ctenophores were closer to the surface. There were also some personal mistakes that I made, such as dropping some of the vials, forgetting to put megalopae in the vials. And there were also some questionable species that we didn't know whether it was euchre or not, and I will show you on the next slide that I have a gel that shows how we were wondering whether it was a euchre species or not, because some of them ended up being spider crabs, which kind of look like euchre megalopae, but they have a slight difference. So as you can see in the bottom right, there aren't as many bands as the rest of the gel, which this is where we were proven right that these were not euchre species that I was doing research on in this certain experiment. So future studies, there's a lot that could be done with this. There could be more research over the next years that could be done with just different variables. You could use different species in the water, different salinities, different weights of the different species, like instead of 25 grams, upping it or lowering it. It could also be done at different times of the year. And yeah, that is a picture of my research group. We all had different projects from the marine lab, and that is the end of my presentation. I hope you enjoyed it. Thank you so much for watching. Have a great day.